Finally, we're going to get to the light reactions. Even though this lecture was called Light Reactions, we spent a bunch of time talking about other things, but now let's get into it. All right, let's talk about where this is occurring before we get into the actual reactions. So I guess you got to wait a little longer. So these are a couple images or cross sections of a C3 leaf. We'll get into what C3 versus C4 is a little bit later. You can see the picture on the bottom is an actual cross section of a leaf, and this picture in the upper left is a cartoon or drawing of a leaf. So there's a few aspects of the leaf I want you to be familiar with, or become familiar with. So one, we need to know the epidermal layers. So that's much like our skin. So this outer layer of cells, you can see these are individual slightly elongated cells here on the top of this leaf. That's an epidermal layer, and then it's got a second epidermal layer here at the bottom. You can see that on the cartoon as well as two epidermal layers. Then all of the area between the epidermal layers is referred to as the mesophyll. And we generally have two types of mesophyll. We have this palisade layer here, which is typically on the upper surface of the leaf. And this is generally characterized by having these really elongated cells all packed together. You can see in this cartoon as well, the cells at the top are more elongated and all packed together. Then the spongy mesophyll is typically at the lower surface of the leaf. And here, this is kind of like the bottom half of this leaf is mostly spongy mesophyll. There's a lot of air spaces, so where my cursor is right now, that's just airspace. And then you can see these more circular cells spaced out throughout the spongy mesophyll. So you can see that again on the cartoon, more air spaces in the mesophyll. And then these are hard to see on the actual cross section, but there are some vascular bundles. They're kind of smeared in the images here. All of these ones that look like they're blurry are the vascular bundles. You can see that better in the cartoon. These are the vascular bundles. That will have, we'll come to that in a second. We've got xylem is pink in this cartoon, and that's where water is being transported. The phloem is transporting mainly sugars back and forth throughout the plant. We'll talk about that in a few weeks. A couple other things to note. You can see a stoma down at the bottom here of this leaf, and the guard cells are the two cells on either side of the stoma. We'll talk a little more about those guard cells and stoma in future lectures, but that's where CO2 is diffusing into the leaf, and then at the same time water is diffusing out. So those are really important apertures or openings in the epidermal layer that we'll be talking about throughout the semester as well. Okay, so back to the palisade layer for a second. The palisade layer has, well, you can see lots of cells have these little green dots in them, and they tend to be more concentrated in the palisade layer, although you can't really see it in the cartoon. You can kind of see it in this cross section. You see these big black dots and more kind of pinkish material in these cells, where more of these cells in the spongy mesophyll are just open and white. Those dark green dots are chloroplasts. Those are subcellular components where the photosynthetic activity is really happening. So photosynthesis is happening primarily in the palisade cells. That's part of the reason why they're in the upper surface, is because they're right where the light is actually hitting the leaf surface and is then going to penetrate right into those cells. And so then the chloroplasts are arranged so that they're in those palisade layers primarily. They're in the other cells as well, but these are where they're concentrated. All right. Here's just, this is a scanning electron micrograph, or SEM. So this is just a really cool one of an actual leaf. So you can see this kind of more lime green-ish, well, maybe that's not the right word, I'm not going to get into the shades of green. So you can see this upper layer. These are those elongated cells that are in the palisade parenchyma. Down here is the spongy mesophyll. Right in the middle of this image, kind of you can see a vascular bundle. So that's, again, where the xylem and phloem is. So that's just a cool way of visualizing where these things are happening in the leaf. All right, so here is a picture on the left, a cartoon of a plant cell. And so you can see the chloroplast located on the edge of the cell. We've got some other stuff that we won't talk about just now. On the right-hand side is a cartoon of a chloroplast, and then the bottom right one is another scanning electron micrograph of a chloroplast. Just a couple things to point out here. You don't have to remember all of these terms. I'll highlight at the end the things I want you to know. What I really want you to remember is the thylakoid membrane. So in this cartoon, these stacks of material here, those are all surrounded by thylakoid membranes. All right, so all of this material are membranes within the chloroplast. And that's really important. I will just highlight, I'm really sorry about that. I want to highlight these stacks referred to as granum, and you don't have to remember that for anything down the road, but I wanted to just point it out for now. Granum are here, and then we have the stroma is in between the stacks of granum. Okay, I'll point out where some things are happening relative to the granum and the stroma here in just a couple minutes. All right, so important component is that thylakoid membrane. So all of the components of the light reactions that we're going to talk about here in just a second are embedded in this membrane. That's really important because membranes are fairly sensitive to environmental conditions. And so water limitation or temperature extremes in particular really affect the ability of a membrane to stay structurally sound or robust. And so if the membrane is damaged or not as stable as you would want it to be, those components of the light reaction start moving around and they can't function as well as they should. So anything that's affecting a membrane in a plant is affecting the ability of the plants to harvest sunlight and convert it to chemical energy. So that's why I really wanted to highlight and have you remember the thylakoid membrane because of its importance. Okay, there are kind of 4 subunits of photosynthesis and then one extra component that's like really should be included but isn't really. So I'm going to talk about them all as kind of one system, but the 4 subunits that are often referred to in photosynthesis are the antenna or the set of pigments. There's 2 different photosystems, so this is the pigments are one subunit. The photosystems are 2 different subunits, and then the cytochrome B6F complex, which we'll touch on briefly, but I don't want you to have to remember that. Those are the 4 subsystems, and then this ATP synthase is another subunit that really can't be separated from all these others. So we're going to talk about them all together. Okay, so general process of the light reactions. Let me, the slides weren't as I expected. Hold on. Okay, I have 2 that I wanted to show you, and I put them, I'm going to pause this and come back in a second. Okay, I'm back. So the general process of the light reactions are kind of put together in what's referred to as a Z-scheme. And it's because the way we think about this kind of results in a Z shape. So if you kind of follow me here, if we start at the bottom here, this photosystem 2, and go up, and then slowly back down and then up again, that gives us a Z, or some people just refer to it as a zigzag pattern. So we'll talk about why it's in the shape of the Z in just a second, but that's the general name of the scheme. I'm going to go to this one real quick and then back. This is another way of visualizing the Z scheme. So we would start here over where you see the green subunit here, which is photosystem 2. We would go up, down through here, back up. And then you could come back down and go up again if you wanted to. So that would be the Z scheme. I'm just trying to show you the Z scheme, basically. The other thing I want to point out with this image, though, is it shows you these different components embedded in this membrane. So the circles and the little legs here, that's a lipid bilayer. So that's the membrane of all plant cells, and lots of organelles inside the cells have this lipid bilayer membrane. So all of them are embedded in that membrane. All right, let's go back to the Z scheme real quick and talk about what's going on. Okay, we've got two different photosystems. We'll talk about a little bit throughout this process. I've got a summary slide at the end, so let's wait to talk about them in more detail then. But in general, what happens is with each photosystem, there are a group of pigments that act like an antenna. And those antenna and pigments are the first thing that captures light energy and then harvests it into that photosystem. And the process that happens is that the electrons in those pigments get excited to a higher state, if you remember back to your basic chemistry classes. And when the electrons are excited, they're really unstable. And so in order to kind of harness that energy before it flies off and becomes fluorescence or something else happens or released as heat, those pigments pass on that excited electron and that energy to a different set of pigments. And so this first part of the Z that goes up are those pigments being excited and entering a higher electron state. And then those electrons get passed down, and that's why you have this series of steps going back down before it goes up again. So it's all about electron transfer. Photosystem 2 has its set of pigments that are associated with it. They pass that electron down to that main photosystem where it produces some products that are important for photosynthesis. We'll talk about in a second. And then photosystem 1 gets excited. It can be excited on its own, or it can be excited from electrons passed down from photosystem 2, and then that whole process goes on again. And then the process of harvesting or harnessing those the energy from the excited electrons results in different products. Again, we'll talk about those in a second. So that's the general scheme. It's why it's a Z, because we're talking about the excitation of electrons and then passing those electrons off. And when you get past them off, they go to a slightly lower energy state, and that's why it's coming back down. So we're losing some energy as we pass it down between all of these different components, but it's important to pass them down, or else that energy will be released as heat or fluoresced as a different wavelength. But by passing it down, it helps it be more stable and gets to a point where the plants can actually produce some usable product. All right? So what I really want you to remember is the Z scheme, and that it's an electron transport process. That's the handing down of electrons that harnesses that energy. I'll point out why we're here. I think I'm jumping ahead of my slides. I've got some of this information later, but we'll get to it. The cytochrome B6 complex that I talked about before, that's right here in the middle. That's important because once the electron gets there, it's pretty much stable. And so that's a point of kind of no return. If the plant can get the energy to that point, then it's locked away and it can use it for future reactions. Up to then, it's a little bit risky in whether it's going to harness that energy or it could be released as heat or fluorescence. Okay, so here's kind of those steps I just talked about. First, those pigments are capturing the light energy. Remember, we refer to those as antenna. The solar energy raises the state of electrons. They pass it on to different pigments. I gave some details on the types of pigments. And then it gets into those reaction centers, so either photosystem 1 or photosystem 2. Something that's important to recognize from an ecophysiological standpoint as we're thinking about differences in plants, is that the proportion of pigments and the structure of those antennae differ between species. So some species might have a different set of pigments or a different proportion of pigments to capture the light quality in their area, whereas a different species might have a different set of pigments in order to best harness the energy from the sunlight that hits those leaves in a particular area or time. Sorry, I shouldn't be standing right now. Okay. All right, that's my cut. Okay, so here I've got kind of a text version of what I just described, and there's some details in here we'll just talk about briefly. So the pigments are that first step in the light energy reactions where the light is putting those electrons into a higher electron state. Then it's handing those electrons off to other types of pigments or molecules that help that energy become more stable. The first set of pigments is referred to as carotenoids, and then the carotenoids pass the electrons off to chlorophyll A and B, and then the chlorophyll passes it to the reaction center. When I talked about photosystem 1 and 2, those are the two reaction centers. Something that's important here for us to think about from a kind of an ecological standpoint is that the types of pigments and the proportion of the pigments differs between species or can differ between species, and that allows species to kind of adjust or acclimate to different qualities of light, and so they can kind of maximize the ability to capture light energy depending on what wavelengths they're actually receiving. The reaction centers, photosystem 1 and 2, are kind of where those electrons end up. Photosystem 1 was discovered first, that's why it's called photosystem 1, but it actually happens second in the process. So that's why it's a little confusing, and that's why it was further right in that diagram. It reacts most strongly to wavelengths of 700 nanometers. Photosystem 2 reacts at 600 nanometers. And because we're doing electron transport and we're losing energy, remember that shorter wavelengths have more energy, so that's why photosystem 2 comes first. And then as it loses some energy and passes that energy off to other molecules, then it can be used at 700 nanometers by photosystem 1. Again, these reaction centers produce slightly different products. So we'll talk about, let me see, I guess we have them here. Okay, so in this system, in our Z scheme here, so you can see in the middle, right at cytochrome B6, we've got this movement of hydrogen ions from the stroma, which we looked at in that image, outside of the thylakoid membrane. So the movement of those hydrogen ions is really important because, it drives the formation of ATP, which we'll talk about here in just a second. ATP, though, is a form of chemical energy, so this stores energy. So photosystem 1 is producing primarily ATP. Then you see the product on the far right here, which is NADPH. This is another form of energy storage or chemical energy storage in plants, and that's produced primarily from photosystem 1. All right, so those are the different products they produce. Just a couple notes on those energy products. I do not expect you to remember the full names of these, but I would like you to remember those acronyms. So ATP, NADPH, NADH, all three of those are really important chemicals, like storage centers, if you will. They're really small, they don't last long, they typically get moved and used somewhere else fairly quickly, but they're really important in kind of storing and harnessing energy. And down here is kind of, they release energy when they're broken apart. So the ATP, the T stands for tri, and the P is phosphate. So when you break one of those phosphates out, you result in ADP and inorganic phosphorus. Or if you break those hydrogen ions off of NADPH or NADH, that releases energy. So that's the process of releasing the energy. Oh gosh, this is going to be driving you crazy, this back and forth. I will get this figured out for next set of lectures. Okay, so those are the forms of energy. Remember those. We'll be talking about those throughout the next couple of weeks. One other thing to be aware of. So solar energy is really, really intense, especially for tiny little, molecular processes inside a plant. And so that amount of energy from the sun can do a lot of damage. And so excess light energy can cause damage to that machinery. So we talked about all of those different subunits, photosystem 1 and 2, and the antenna, and the B6 complex, and the ATPase synthase. It can damage those things. It can damage the membrane. Little bits of damage can be overcome fairly quickly, but if that really high intensity light persists for very long, then you get damage to those components that cause a reduction in photosynthesis. And so photoinhibition in really places where you get high intensity of sunlight. So hot places often get it, also high elevation when it's cold and you get high light environments. Those are places where we see lots of photoinhibition. Photosystem 2, which we talked about, is a little more susceptible, and in the coming weeks we'll be talking more specifically about photosystem 2 and that sensitivity. All right, so just a few things that I want you to keep in mind. So light reactions are harnessing the solar energy into those chemical bonds, which is ATP, NADPH, NADH. The rate limiting step is the electron transport, not the pigments harnessing the light, but it's passing those electrons off is the slowest part of this process. Pigments and enzymes involved require a lot of nitrogen and maintenance. And so when we were talking about carbon budgets and maintenance being part of that, maintaining these enzymes is fairly expensive for plants. You got those 3 forms of chemical energy, that Z-scheme. Know what that Z-scheme is, and know that the shape of that Z-scheme is a result of exciting electrons and then having those passed off, and that energy being lowered at each step in that process. That thylakoid membrane, remember that's important because anything that's going to affect membrane stability is going to affect photosynthetic rates. And then just know generally what photoinhibition is, that it's damage to the photosynthetic machinery, if you will, and can slow down photosynthetic rates. All right, so that's what I want to you to remember from that section. One thing here, I might have said this during our first meeting, but this part of the class, well, these lecture components of the class will be a little bit towards the spectrum of plant physiology. So we talked about, ecophysiology as being kind of a bridge between physiology and ecology. And so sometimes to understand these mechanisms, we have to go to the physiological side, but then through our readings and our labs, hopefully we'll go a little more towards the ecology side. So hang on during these processes and these lectures because they're really important to understand some of these mechanisms. And it will help us interpret the papers that we're going to be reading and the data we're going to be collecting throughout the class. All right. Oh, I have one other slide. One thing that I want to try to do at the end of these little lectures is, because of what I just said, tends to be on the side of physiology. I want to bring it back a little bit so we can think about what things we talked about that are going to be related to a larger scale or more ecophysiology. So a couple of things here. The interspecific differences are always going to be really interesting and important. A couple of those areas that might differ between species, I mentioned the pigments or the antennas can differ between species in their ability to capture different types of light. And then also the amount of resources a plant invests in the chloroplasts in that palisade layer. And that's going to differ a lot depending on the light environment a plant might grow at. Yeah, we'll leave it at that for now. Damage to the membrane. So this first one's talking about species differences. Thinking about damage to the membrane, this is more like a seasonal thing within an individual species. How might a cold a late spring cold snap affect the membrane, or how would a heat wave affect the membrane of a species? Well, it's going to definitely affect that membrane to some degree, and that will impact photosynthetic rates and growth. So that's important to keep in mind. And then adjustments based on the light environment. So as an environment, the light environment changes, plants can acclimate to some degree to those changes, and that might cause some changes in the behavior of that plant over time. So those are some slightly bigger picture things or bigger scale things that these light reactions relate to. All right, now we're done for today.